Hello, everyone. This is for those of you who aren't able to attend my talk. So without further ado, hello, my name is Chaz, and today we are going to be comparing molecular and morphological methods for distinguishing introduced Prussian carp from goldfish here in Alberta. So Prussian carp are a freshwater cyprinid species native from Central Europe to Asia. They are otherwise considered invasive in regions throughout Europe and the Middle East. They've recently reached introduced status here in Canada and are widespread in Alberta's main river drainages and basins. Given how difficult it is to control an introduced species population after it's been well established, their early detection is the best strategy for the mitigation of their potentially negative impacts on native populations, ecosystems, and economies. While several characters listed here contribute to the species success as an invasive, the most remarkable factor is their ability to reproduce through gynogenesis. Now, through gynogenesis, Prussian carp are able to parasitize the sperm of another cyprinid fish species in order to produce clonal offspring. In this respect, all it takes is one female Prussian carp in a freshwater body of water where other cyprinids are present in order to produce a potentially dominating population. Now, a suspected population has been reported recently in Mahogany Lake, which is an artificial freshwater lake, community freshwater lake down here in Calgary. Now, the community's primary concern is the population's potential impact on their trout stocks, which is replenished annually for recreational fishing. There's also a question as to how these fish got here when this is an artificial lake not connected to any surrounding bodies of water. Now, what's difficult with the early detection of Prussian carp is that they look a whole lot like wild-type goldfish, which is another introduced species here in Canada. While both species are ecologically similar, goldfish lack the ability to reproduce through gynogenesis. While there's been debate over their taxonomic placement, current research suggests that the two species are sister taxa under the same genus Carassius. Now, one way we can identify specimens of an unknown species is through DNA barcoding. Just like how we can identify a can of corn using its barcode, we can identify species using special DNA barcodes. Now, DNA barcodes are particular regions of DNA that show little intraspecific variability, but significantly greater interspecific variability. Through DNA barcoding, we can take a query sequence from an unknown specimen and compare it with reference sequences of known species in order to determine the closest species match. Now, this was the method used in this 2014 study, which first confirmed Prussian carp in North America. So for our collection sites, on the left, of course, we have Mahogany Lake, but we also have some fish donated to us by Trout Unlimited Canada in the Carseland Bull River Headworks Canal. These fish are relevant to our study because if the fish from both sites do end up being the same species, it could give us some hints as to the origins of the population in Mahogany Lake, especially given the two sites' close proximity to the Bull River, around which Prussian carp has been reported. For DNA barcoding, we took fin clips from all of our samples, put them in these special live scanner kits. Our samples were then sent to the University of Guelph for sequencing, and those sequences were then uploaded to the Barcode of Life Data Systems database to be compared with their reference sequences of known species. Species match is designated to those query sequences that show a less than 1% sequence divergence from reference sequences of known species. Our results were interesting in that all of our sequences from both sites were genetically identical, which indicates that all of our fish belong to the same species. However, DNA barcoding results was conclusive for 5 different designated species in Bold's reference library, suggesting that our fish could be Prussian carp, goldfish, Prussian carp, a goldfish subspecies, or a Carassius hybrid. Since DNA barcoding wasn't helpful in helping us pinpoint the exact species that our fish belong to, we thought that morphology could give us a better idea. So our morphological analysis of our samples was based on these 7 characters described in the Handbook of European Freshwater Fishes for distinguishing between Carassius species. For our study, we looked at expected values for Prussian carp, goldfish, and Prussian carp. The first character we looked at was serrations on the last simple dorsal and anal fin rays. Now for Prussian carp, we expect these serrations to be weakly serrated, while for goldfish and Prussian carp, we expect these fin rays to be strongly serrated. The second character we looked at was branched anal fin ray count. Now for Prussian carp, we expect to find 6.5 branched anal fin rays, while for goldfish and Prussian carp, we expect to find 5.5. Now, anal fin rays count as 1.5 fin rays when they branch from the same origin. So that's kind of weird, but we'll take it. The third character we looked at was peritoneum color. Peritoneum is the lining of the abdominal cavity in vertebrates. For Prussian carp, we expect this to be a whitish color. However, for goldfish and Prussian carp, we expect this to be a black color. For our fourth character, we looked at total lateral line scales. These include those scales that follow the lateral line all the way to the caudal fin. In Prussian carp, we expect it to range from 31 to 36 in the upper ranges, while for goldfish and Prussian carp, we expect this to be in the lower ranges. As you can see, for goldfish and Prussian carp, their ranges overlap by a substantial amount. Similarly, for gill raker count, we have expected values in the lower ranges for Prussian carp, while our expected values for goldfish and Prussian carp were in the upper ranges with significant overlap between those two species as well. For our sixth character, we looked at body color, which is expected to be golden green in Prussian carp, golden brown in goldfish, and silvery brown in Prussian carp. Lastly, for our seventh character, we looked at the shape of the free edge of the dorsal fin, which is expected to be convex in both Prussian carp and goldfish, and is otherwise expected to be concave or straight in Prussian carp. Now, here in blue, we have the values for those confirmed Prussian carp in that 2014 study. In yellow, we have the values for our fish. And as we can see, our fish overall are more morphologically similar to Prussian carp than either Prussian carp or goldfish, based on these 7 characters from the Handbook of European Freshwater Fishes. This confirms that our specimens are indeed Prussian carp. However, this still begs the question as to why our DNA barcoding results give us a conclusive species match for 5 different designated species in their library, which leads us to another question. Are there mislabeled sequences in BOLD's library? This led us to a couple specific predictions. The first prediction is that those identical sequences in BOLD's library that aren't Prussian carp are a minority in their respective species group, and that BOLD's reference sequences for both goldfish and Prussian carp will have a significantly greater mean number of variable sites than sequences for Prussian carp when compared to our fish. So to determine the number of variable sites that each of BOLD's publicly available sequences for those 3 species have compared to our query sequence, all we had to do was take our longest sequence from Mahogany Lake, which is 521 base pairs long, and align it using genetics alignment software, align it with sequences for goldfish, Prussian carp, and Prussian carp that were publicly available in BOLD's reference library in order to find those variable sites. So here we have the total number of sequences that we could find for each species versus the total number of identical sequences in those species groups that were identical with our query fish. And what we find is identical sequences were indeed a minority for all 3 species, for Prussian carp and goldfish, and even in Prussian carp. As for our second prediction, here we have the distribution of sequences based on number of variable sites for each species group. As you can see, for goldfish, most sequences diverged from our mahogany lake fish at 4 variable sites, while for Prussian carp, most sequences diverged from our mahogany lake fish at 33 variable sites. Both of these values, sorry about the noise, both of these values are not much, but both of these values turned out to be greater than the value for Prussian carp, where most sequences varied from our mahogany lake fish at only 1 variable site. When looking at this interval plot, we can see that the means for both goldfish and Prussian carp are greater than the mean for Prussian carp. Since we're dealing with differently shaped distributions, unequal variances, and very different sample sizes, we chose to do a Welch's ANOVA to compare means between the 3 species groups. And our Welch's ANOVA results showed us that there is a significant difference within the groups, and our post hoc tests confirm our second prediction, in that sequences for both goldfish and Prussian carp had a greater mean number of variable sites than sequences for Prussian carp when compared to our mahogany-like fish. So in summary, morphology confirms that our samples are indeed Prussian carp. Bold's results, our DNA barcoding results, were inconclusive, likely because of some mislabeled Prussian carp in Bold's library that were labeled as Prussian carp or goldfish. Otherwise, our fish are most genetically similar to Prussian carp than those 2 fishes when using statistics. These evidences confirm that Prussian carp are present in both Mahogany Lake and CBR-HC. So I'd like to thank these individuals and groups for their contributions, and I'd actually like to read through it. I would like to thank the following: my supervisor, Dr. Matthew Morris, for his continued support and encouragement throughout the course of this research project and throughout the course of my undergraduate studies. Dr. Ted Pike for supervising my many hours in the lab. John Wiest and Abigail Fuca for their statistical guidance. Jesse Garcia, Aaron Weberg, and my dad, Roel Comia, for their help in identifying and processing fish for DNA barcoding. Trout Unlimited Canada for their effort in capturing half the fish I used for this study. And the community of Mahogany for donating the other half of the fish that I used in this study. Mahogany Homeowners Association for allowing me to do science in their beautiful community lake. Natasha Pentiluk for insight regarding current research on Prussian carp in Alberta, and to all my other friends and family who contributed to this project. Thank you very much. So here are some key resources that if you want to pause here and jot these down. These are particularly concerning Prussian carp here in North America. So those would be great reads. I think they're free. If you have any questions, you know where to message me. Otherwise, you can use this email. And that's that. If you want to stick around, I'm going to go over some extra tidbits that weren't included in the talk that I did today. If you're confused with the concept of variable sites, as you know, DNA is made up of amino acids. Is that right? No. DNA is made up of these nucleotides, adenine, cytosine, tyrosine, and guanine, and they are connected to each other in a chain. And well, when our samples get sequenced, they come out in this really long chain of nucleotides, and we get the data in these letters. So here we have an unknown sequence of 8 base pairs long with this nucleotide configuration. We don't know the species of the sequence. So what we're going to do is we are going to align it with sequences from known species, particularly species A, species B, and species C. So right off the bat, you can tell that known species, our query sequence looks exactly like species A. And this leads us to think that our unknown sequence might be the exact same species as species A. For species B, however, we can see at site number 6 that there's a mutation where the guanine is replaced with the cytosine. And we can translate that as this sequence having one variable site from our unknown sequence. This is such that the sequence for species A is more similar than species B, more similar to our query sequence than species B. And then looking at species C, we can see that there are 2 variable sites, showing an even greater divergence or dissimilarity from our unknown sequence. So when looking at this data, we can tell that our fish is most likely the same species as species A, and less likely to be the same species as species B and species C. So another tidbit was that using our longest sequence, we were able to tell that mean number of variable sites was significantly greater in sequences for goldfish and Prussian carp than sequences for Prussian carp when compared to our mahogany-like fish. This data shows that this was also the case using our longest sequence from CBRHC. So this is just our due diligence showing that fish from both sites are more genetically similar to Prussian carp. Now, it is no surprise that a population has been well established in mahogany, because during our site visits, we found that the conditions were ripe for a Prussian carp population to boom. Here we have fathead minnows, which are a cyprinid species. Now, as previously mentioned, Prussian carp are able to reproduce through gynogenesis, in which they parasitize the sperm of other cyprinid species. So all it took was one female to be introduced in mahogany-like to parasitize the sperm of the fathead minnows that are already present there in order to produce an entire population of Prussian carp. No sexual reproduction required. Furthermore, there was marked algal growth in the lake. And Prussian carp, I found a paper about Prussian carp diet, which revealed that filamentous algae was a primary component in the Prussian carp diet. There was also crustaceans in the lake, as you can see here, the larvae of which also make up part of the diet for Prussian carp. So as we can see, the conditions are pretty ripe for, just by looking at these evidences. There are other factors about the lake that are conducive to Prussian carp population growth. Here's a neat figure that unfortunately I couldn't include in the talk. And this is showing fork length, which is essentially the size of the fish, in relation to gill raker number. Now, going back, if we look at gill raker count, we can see that there is much overlap in the expected values for goldfish and Prussian carp. However, the upper limits for Prussian carp were above the upper limit for goldfish, with 37 to 52 for Prussian carp and 38 to 47. Now, a few of our fish, particularly our small fish, had gill raker counts that were well within the range for goldfish. So this could raise the possibility that our small fish are actually goldfish, ignoring all of our other evidences. But what was interesting is that most of our small fish were from CBRHC. The fish from CBRHC were much smaller than the fish that we collected from Mahogany Lake, and this wasn't really random sampling. So that does raise the possibility that they belong to two different species. However, when looking at this graph, we can see that some of our fish from Mahogany Lake, denoted by the blue triangles, are actually in the size range of those fish from CBRHC, denoted by the red circles. Those fish that were in the same size range also tended to have the same gill raker counts. And here we can see that there's a significant linear increase in gill raker count with fork length. This suggests that our small fish in CBRHC are actually just those juvenile Prussian carp that are on their way to having the higher gill raker counts, which is more in line with what is expected in Prussian carp, but they're just small. I haven't been able to find any literature that gill raker count increases with size in fish, but this is some pretty good evidence that it does, especially since all of our fish were genetically identical. So thank you very much.